Hello, everyone. My name is James. Welcome to our monthly webinar. Today it's my pleasure to introduce Dr. Li. Will give us a talk for his research. Dr. Li is a principal investigator at Mass General Hospital and also assistant professor at Harvard Medical School. He completed his Ph.D. at the University of Hong Kong and his postdoc work at Mass General Hospital. His lab at the Department of Anesthesia at the Mass General Hospital focused on the non-coding RNA and RNA modification in cardiac and cerebral response to exercise in diabetes and aging. Today, Dr. Li will give us a talk about epitranscriptional regulation of cardiac response to exercise. So we have a webinar followed by the Q&A section. So if you have any question during the talk, you can type your question in the Q&A section. And also after the talk, we will open our Q&A section. You can raise your hand. I will give you the access. So Dr. Li, the time is yours. Thank you.
Thanks, Jen, for the kind introductions. Hope you can see my screen. Okay, yeah, thank you again for the kind introductions and the invitations to share our work here. Here are a few disclosures. So as Jens mentions, our lab is primarily focused on two areas, diabetes and aging, if we can call aging a disease. I cannot emphasize enough that the prevalence of diabetes is alarming, with a risk that doubling globally since 1990s. And similarly for aging, the number of people aged over 60 years is projected to be 5 times that of 1950s. Importantly, most patients with diabetes in older adults, the major cause of death is cardiovascular disease. Therefore, there are unmet clinical needs for therapeutics for cardiovascular disease in these populations. Our lab has long been interested in studying the heart. We're trying to understand the molecular mechanism driving metabolic and age-related cardiac disease, and try to understand the cardiac benefit of exercise in this context. Multiple process and molecular mechanism has been proposed to contribute to these conditions. We are particularly interested in circulation factors, noncoding RNAs, and epigenetic changes at the RNA level, the so-called RNA modifications. RNA modifications are chemical changes that occurs on RNA molecules after transcriptions. Today, there are more than 100 RNA modifications has been identified. And different enzymes that regulate these RNA modifications has been demonstrated to regulate various aspects of cardiac physiology. However, their roles in diabetic cardiomyopathy or cardiac metabolic disease has less been reported, and their roles in cardiac benefit of exercise in this context has never been studied. Our work in the lab is mostly focused on RNA acetylations, AC4C, and RNA modification, methylations, M6A. I will quickly go through one project here. So M6A is a dynamic RNA methylation and demethylation process that involve writers that install M6A modifications. Erasers that remove these modifications, and readers that recognize this M6A modified RNA. To examine the role of M6A in the exercise benefit of diabetic cardiomyopathy, we use a combination of high-fat diets and a low-dose steroidozosin STZ to induce type 2 diabetes in mice, and some of these mice were subjected to treadmill running for 8 weeks. compared with chow-fed sedentary mice and diabetic mice that was sedentary has impaired systolic and diastolic functions, showing here the blue bar that reduce fractional shortening, which indicates how well the heart pumps blood up, and increase E to E pi ratios indicates how well the heart fills with blood. And as expected, at the end of 8 weeks running, we found that exercise improved both systolic and diastolic functions in diabetic mice, and no obvious changes observed in systolic and diastolic functions in chow-fed mice after exercise. We then looked into the changes of M6A in this mouse heart. We found that total cardiac M6A levels were decreased in diabetic mice and increased by exercise in both chow and diabetic mice. To look for more specific M6A regulations, we measure the changes of some key M6A regulators. Among those, only MESO3 methyltransferase of M6A that dynamically changes in diabetes and exercise, where cardiac MESO3 was downregulated in diabetic hearts and increased by exercise both in the chow and diabetes mice, consistent with the changes that we saw for M6A change in this mouse. Importantly, similar to what we saw in diabetic mice, cardiac MESO3 mRNA and protein expressions were reduced in patients with dilated cardiomyopathy, both with or without diabetes, and more downregulates in those patients with diabetes compared to those without. Expression level was positively correlated with left ventricular ejection fractions, although this is mostly driven by the low expression level of MESO3 in diabetic hearts. So taken together, this data suggests that exercise may protect a diabetic heart through MESO3. To examine the role of MESO3 in diabetic cardiomyopathy and exercise, we sought Gene Biotech's help to construct a MESO3 Brock's mice line where we insert lost sign flanking exosome 2 to 4 of the MESO3 loci. When we inject AAV9 encoding a cardiomyocyte-specific promoter troponin T-driven pre-recombinant into this mice, we can efficiently knock out MESO3 in cardiomyocytes. Again, we use high-fat diet and STZ to induce diabetes in these flux mice. 5 weeks after STZ injections, or 1 week before exercise, we inject AAV pre into these mice, assuming that AAV needs at least 1 week to express genes at detectable level. So in this way, we can test the role of MESO3 in exercise after its initiations. So mice will then run for 8 weeks, and we found that in sedentary mice, cardiac-specific MESO3 knockout did not affect systolic and diastolic functions, but it cancels exercise effect both in systolic and diastolic function, showing here the blue bars that reduce fractional shortening and increase E to E pi ratios. So altogether, this results demonstrated that exercise is effective in attenuate diabetic cardiomyopathy, and that cardiomyocytes MESO3 is essential for this process. So over the years, we and others have shown that virtually every pathway identified as functionally important in the cardiac response to exercise protects the heart against pathological stress when we mimic the change seen in exercise. We have identified a series of these targets, both non-coding RNA or coding genes. So we then want to test whether overexpression of MESO3 can mimic that we saw in exercise that protects the heart in diabetes. Again, we use AAV9 with troponin promoter to induce MESO3 in cardiomyocytes. AAV9 was injected 5 weeks after STZ injections, at a time that diabetic mice already developed systolic and diastolic dysfunctions. And in this, similar to what we saw in exercise, overexpression of MESO3 improve both systolic and diastolic functions in diabetic mice. And acknowledge that there's no perfect animal models for diabetic cardiomyopathy, so we also test this in DBDB diabetic animal mice. AAV was injected at 16 weeks of age, an age at which these mice have developed systolic dysfunctions. And we saw a similar protected effect with MESO3 overexpression, showing here the blue bars compared to the red bars that injected with AAV control. And some people may concern about AAV. So in addition to AAV to induce MESO3 in hearts, we also test small molecules for MP3C that specifically targeting the MESO3, MESO14, and the WT8P complex, which catalyze the formation of M6A. We found that injection of MP3C was effective in improving fractional shortening and E to E prime ratios as early as 2 weeks after injections. So altogether, these results demonstrated that inductions or activations of MESO3 is sufficient to protect against diabetic cardiomyopathy. Now look into the mechanism. We start with the upstream, looking at exercise. How exercise increase MESO3 in diabetic hearts. We focus on transcription mechanism by looking at what transcription factors that bind to the promoter region of MESO3. We compare this transcription factor from mouse and human genome and found 5 conserved transcription factors that potentially bind to MESO3 promoters. We then focus on STAT3, since our previous study showed that STAT3 activations is cardioprotective in myocardial ischemic reperfusions in diabetic hearts. There are 2 STAT3 binding sites on MESO3 promoter regions. We then perform cut and run assays using a STAT3 antibody to pull down common fragments, and we found that the binding fragments containing STAT3 binding motif in MESO3 was reduced in diabetic hearts here, an increase by exercise both in the child fat mice and the diabetes mice. And then in neonatal cardiomyocytes, overexpression of STAT3 using a lentivirus increase MESO3 protein expressions, while when we knock down MESO3 by siRNA, that not affect STAT3 protein expressions. So together, this results indicates that exercise increase MESO3 through STAT3 in diabetic hearts. For the downstream mechanism, we perform a bulk RNA sequencing in hearts from child fat and diabetic mice. And in diabetic mice with or without MESO3 overexpressions. And we also compare this to M6A immunoprecipitation sequencing in diabetic hearts, and we identify YBX1 white box binding protein 1, which was downregulated in diabetic hearts, but was increased by MESO3 in both trial and diabetic hearts. And similar to the changes of MESO3, cardiac YBX1 protein expressions was downregulated in patients with dilated cardiomyopathy with or without diabetes. And to examine whether MESO3 regulates YBX1 through M6A, we fuse that Cas13 with MESO3, and in combination with RNA that targeting N6A binding sites on YBX1 in cardiomyocytes, we found that transfection of this two lentivirus was able to increase YBX1 protein expressions. These results suggest that YBX1 works downstream of MESO3 in diabetic hearts, and that MESO3 regulates YBX1 is an M6A dependence. So to further gain insights into how MESO3 regulates diabetic cardiomyopathy, we compare differentially expressed genes that identifies by RNA sequencing of heart from diabetic mice with or without MESO3 overexpressions. Not surprising that these genes were enriched in pathways that related to oxidative stress. And in this, in mice, we found that our productions are measured by DHE staining that was increased in diabetic mice and reduced by MESO3 overexpressions. This result suggests that MESO3 protects diabetic cardiomyopathy through reducing oxidative stress. So now the question is, how and what is the role of YBX1 here? Interestingly, a previous study showed that YBX1 can bind to the 5-UTR region of MRF2, a potent anti-oxidative genes that promotes the translation of NRF2 in sarcomal cells. So in isolated cardiomyocytes, we perform polysome fractionation assays and found that overexpression of YBX1 increased NRF2 mRNA expression in the high molecular weight polysome, suggesting that YBX1 increased NRF2 translation in cardiomyocytes. We also found that overexpression of YBX1 increased nuclear NRF2 protein expressions, and also increased NRF2 downstream effectors, HO1 and NQO1. So this suggests that YBX1 regulates oxidative stress through NRF2. Now the question is whether YBX1 mediates MESO3's effect in diabetic hearts through NRF2. So in vivo in diabetic mice, we found that overexpression of MESO3 increase YBX1 and NRF2 activations and improve systolic and diastolic functions. The red showing here the red bars. However, this was reversed when we knocked down YBX1 at the same time. So in here, the blue bars that YBX1 knocked down decrease nuclear NRF2 protein expressions, and also reduce systolic and diastolic functions, improvement induced by MESO3 overexpressions. So taken together, this result suggests that YBX1 works as a downstream mechanism, as a downstream effector of MESO3 in diabetic hearts, and it functions through NRF2 mediated anti-oxidative stress. Last experiment for this project that we found that overexpression of YBX1 in diabetic mice is effective in attenuating diabetic cardiomyopathy, showing here that overexpression of YBX1 increase fractional shortening, decrease E to E pi ratios, and also reduce oxidative stress in the heart. So a quick summary, in diabetic hearts, SAS3 phosphorylation is reduced that reduce meso3 expression, which lead to downregulation of YBX1, results in reduced NRF2 activations, increase oxidative stress that eventually lead to diabetic cardiomyopathy. And exercise, on the other hand, increase SAS3 and then induce meso3 that promotes YBX1, which then promotes NRF2 translation and activations, reduce oxidative stress, and protects against diabetic cardiomyopathy. So this is just a quick walkthrough of the projects, and if you want more details, you can find our recent publications and circulations. And obviously, here there are still a lot of unanswered questions that need further investigation. So, for examples, what is the rule? What is the exact rule of a stress-free in exercise? And also, any M6A readers that involved in this process. So we're working on this, and I hope there will be other chance to talk with you about this. So, what I show you here today is that we add one more targets and meso3 to the cardiac benefit of exercise. And we recently also identified another target, MAT10, which is also for RNA acetylations, AC4C, that also mediate exercise benefits in the hearts. If you are interested, you can find our recent publications at Circulation Research. With that, I would like to thank all the people in the labs, and also want to thank our wonderful collaborators locally and internationally, and also want to thank our funding support. And we are hiring. If you are interested, please feel free to send me emails. I think I will stop here and happy to take any questions.